Hello viewers, today my topic is lipid metabolism. But before going on to the intricacies of this topic, I would like to tell you something regarding the general aspects of metabolism. Now what is metabolism? Metabolism is actually an integrated network of chemical reactions which are catalyzed by allosteric enzymes and involves a coupling between the exergonic and the endergonic reactions. As we can see in this particular slide, the different metabolic reactions are intertwined very tightly within a biological system, and they culminate in a particular common intermediate which is called the acetyl-CoA, which through the successive intermediates of the citric acid cycle, the electron transport chain and the oxidative phosphorylation gets converted to carbon dioxide, water and energy, which is conserved in the form of ATP, the biological energy currency. There are four principal characteristics of a particular metabolic pathway, and these are irreversibility, the presence of a first exergonic committed step, a tight regulation at this particular step, and number 4 is compartmentalization, that is the occurrence of metabolic reactions within particular subcellular compartments in case of eukaryotic cells. Now, metabolism can be classified broadly into two subtypes, the catabolism and the anabolism. Catabolism is actually the degradation of complex macromolecules into their basic building blocks. It is actually bioenergetically a downhill process. There are three successive stages of catabolism as can be shown in this particular slide. The macromolecules like the triglycerides, the glycogen and the proteins are broken down to their respective building blocks, fatty acids, glucose and the amino acids. In the second stage, these intermediates are further metabolized into a few simpler ones including pyruvic acid and acetyl-CoA. After that, in the third stage, the acetyl-CoA with help from citric acid cycle, the electron transport chain and oxidative phosphorylation gets converted to carbon dioxide, water and ATP. The metabolic reactions that actually leads to the biosynthesis of macromolecules are called anabolic reactions. In anabolic reactions, there are also three stages of formation of biological macromolecules, but they happen in the reverse direction as compared to the catabolism. Like for instance, first the precursor molecules are biosynthesized, from then the building blocks are synthesized, and last of all the building blocks are assembled to form the biological macromolecules. As can be shown from this particular slide, the complex metabolites are degraded down to their respective simple products. In this particular process, ADP is phosphorylated to ATP and NADP plus is reduced to NADPH. Now, this particular branch is called catabolism. In case of anabolism, the simple products are assembled together, harnessing the energy released from ATP and NADPH to produce the complex metabolites. This particular slide actually will show us how the catabolic and the anabolic reactions take place within a biological cell, but essentially in the reverse direction. Catabolism and anabolism generally takes place in different subcellular compartments within eukaryotic cells. This is because they have to be independently regulated and they have to made to occur independent of each other simultaneously as well as concurrently. There is a central pathway that is accessible to both the catabolic and the anabolic pathways. This is called the amphibolic pathway, means it can go to the catabolic pathway or it may even go to the anabolic pathway, all dependent on the energy status of a cell. The most important of the lipids are the triacylglycerols, or the tags as they are popularly called. They are famous to us as the fats and oils that we have in our day-to-day diet. They comprise 90% of the total dietary lipids. Now, tags can be stored in the pure and anhydrous form, and they give us an energy release of about 9 kilocalorie per gram. As can be shown in this particular slide, the tags are stored in the cells of the adipose tissue, which are called the adipocytes. A structure of tag is even shown in this diagram, which has a basic backbone of glycerol esterified at its 3 carbon atoms by 3 fatty acids. Tags are water insoluble because lipids, as we all know, are nonpolar, and the digestive enzymes are polar in nature. So essentially, the tags are hydrolyzed, which is called the lipolysis process at the lipid-water interfaces. There are a lot of hormones in our body that regulate lipolysis, which are called the epinephrine, the norepinephrine, the adrenocorticotropic hormone, and the glucagon. On the other hand, insulin inhibits lipolysis. Now, after lipolysis, the tags are actually incorporated into the micelles in order for they getting acted upon by the pancreatic lipases from the external sides of the micelles. Coming to the action of the pancreatic lipases, they actually gets enzymatically activated once they come into contact with the lipid-water interfaces. This is known as interfacial activation. Pancreatic lipases usually cleaves down a triacylglycerol from their carbon number 1 and carbon number 3. The fatty acids are removed sequentially, giving rise to diacylglycerol and monoacylglycerol. The free fatty acids are released into the different tissues, whereas the glycerol molecules are sent to the liver and the kidneys where they are further metabolized. Coming to the action of the phospholipase A2. Phospholipase A2 actually cleaves down the fatty acid from carbon number 2 of any phospholipid, resulting in the release of a lysophospholipid. For example, in this slide, we can see that phospholipase A2 is acting upon phosphatidylcholine and releasing lysophosphatidylcholine together with a fatty acid from carbon number 2. After the digestion of lipids are completed, the digested products have to be absorbed for better metabolic benefits. The lipid digested products are taken up by the micelles in order to facilitate their transport. Also, inside the intestinal lumen, the fatty acids are complexed with a cytoplasmic protein called the intestinal fatty acid binding protein that helps it to be more water soluble and also protects the tissues from their detergent-like activities. The lipids, after their absorption is completed, the digestion products are assembled to form tags once more. And these tags, after they are resynthesized from the lipid digestion products, are taken up by the lipoprotein transport particles called the chylomicrons. And these chylomicrons are then released into the bloodstream by the lymphatic system. In this particular slide, we would see how the chylomicrons are formed. In the intestinal lumen, the triacylglycerols are broken down to fatty acids and monoacylglycerols. In the mucosal cells of the intestine, these fatty acids and monoacylglycerols are assembled to form the tags once more. These tags, together with help from other lipids and lipoproteins, are assembled into chylomicrons and then they are released into the bloodstream. Similarly, the tags which are resynthesized from the lipid digestion products in the liver are assembled within the VLDL, that is the very low density lipoproteins, and they are released directly into our bloodstream. But the tags which are contained in such a fashion within the chylomicrons and the VLDL are acted upon by the membrane-bound lipoprotein lipase in the skeletal muscles as also in the capillaries of the adipose tissue. In this particular slide, we get to see how the triacylglycerols are digested by the lipases with help from 3 water molecules, releasing the basic backbone L-glycerol and the 3 fatty acids. The fatty acids are usually taken up by the skeletal muscle cells and the adipose tissue, whereas the glycerol are transported away to the liver where they are further metabolized. As is shown in this particular slide, we can see that L-glycerol is actually first phosphorylated by glycerol kinase to yield glycerol 3-phosphate. This glycerol 3-phosphate is then oxidized to dihydroxyacetone phosphate, and then this dihydroxyacetone phosphate is further isomerized to glyceraldehyde 3-phosphate, both dihydroxyacetone phosphate and glyceraldehyde 3-phosphate being the glycolytic intermediates. The fatty acids which are released are extremely toxic. So the tissues cannot harbor them for very long, so they have to be conjugated to the serum albumin to facilitate their mobility to the other tissues for beta oxidation. As can be seen in this particular diagram, the lipids are first digested to glycerol and fatty acids. These fatty acids from the tissues enter the bloodstream where they find the serum albumin with which they are tagged, and then they are transported away to some other tissues for their beta oxidation. The beta oxidation process was discovered in 1904 by Franz Knupp. Franz Knupp actually carried out his classic experiment with the even chain and odd chain fatty acids being fed to dogs with their omega or the last carbon atom being labeled to benzene rings. He tried to isolate the phenyl containing metabolic products from the urine of those dogs. As can be seen from this particular slide, the dogs who have been fed with the odd number carbon fatty acids, hippuric acid could be isolated, which is the glycine amide of benzoic acid, whereas the dogs which have been fed with the even chain fatty acids, Franz Knupp could isolate phenyl aceturic acid, which is the glycine amide of phenyl acetic acid. So in this particular slide, it is shown very clearly how beta oxidation takes place with 2 carbon units being removed at each and every turn. Franz Knupp could easily tell the world that fatty acid oxidation takes place at the beta carbon. This slide actually focuses an overview of the fatty acid oxidation at the beta carbon. In stage number 1, the 2 units are successively removed. Here is shown the example of palmitoyl-CoA. After that, the fatty acids are broken down to the most simple products, the acetyl-CoA, which enters the citric acid cycle, gets converted to carbon dioxide, and the electrons that they release enter the electron transport chain or the respiratory chain, and then they are donated to oxygen, that is the terminal electron acceptor, to water. At the same time, ADP is phosphorylated to ATP, the biological energy currency. Now, prior to beta oxidation, the fatty acids have to be activated. The activation of fatty acids takes place on the outer mitochondrial membrane, and the enzyme responsible is known as the fatty acyl-CoA synthetase. This particular slide will show you how fatty acid reacts with the coenzyme A with the participation of ATP releasing AMP, PPI and fatty acyl-CoA. Paul Berg have further elucidated this pathway into 2 steps. First of all, as can be shown in this slide, the fatty acids reacts with ATP molecule to produce fatty acyl adenylate and PPI. Next, this fatty acyl adenylate combines with the coenzyme A to produce acyl-CoA and AMP. This particular slide will show you the basic outline of the fatty acid activation, but one thing must be borne in mind that the delta G naught prime, the standard free energy change under physiological condition for this part of the fatty acid activation is only minus 15 kilojoule per mole. And this particular reaction from the bioenergetic point of view, the delta G naught prime is very close to zero. So to move the reaction forward, we need the hydrolysis of pyrophosphate by inorganic pyrophosphatase enzyme to release 2pi and a delta G naught value of minus 19 kilojoule per mole. So if we now add up these two delta G naught prime values, it is coming to minus 34 kilojoule per mole. That can be shown in this particular slide. The fatty acids reacting with coenzyme A, ATP, and water giving fatty acyl CoA, AMP, inorganic phosphate, and proton with a delta G naught value of minus 34 kilojoule per mole. That is a very good value for the reaction to proceed forward. Now, after the fatty acids are activated, they have to be transported because activation takes place in the outer mitochondrial membrane and the actual oxidation of the fatty acid at the beta carbon takes place in the mitochondrial matrix. So, the fatty acid has to be transported from the outer mitochondrial membrane across the inner mitochondrial membrane to the mitochondrial matrix. So, this needs the help of a shuttle system that can be shown in this particular slide. This is called the fatty acyl CoA transport shuttle and gets completed in 4 steps. In step number 1, the fatty acyl CoA donates the fatty acyl group to carnitine, which is 4-trimethyl amino 3-hydroxybutyrate, a zwitter ionic compound. Carnitine accepting the acyl part of the fatty acyl CoA gets converted to acyl carnitine by the enzyme CAT1 or carnitine acyl transferase 1. This is the rate limiting step of fatty acid oxidation and is inhibited by malonyl CoA. After this, in step number 2, the acyl carnitine gets translocated to the mitochondrial matrix using the translocase enzyme. In the mitochondrial matrix, the acyl carnitine finds the mitochondrial pool of coenzyme A and it gives its acyl part to coenzyme A once more to form the fatty acyl CoA, itself getting converted to free carnitine that in step number 4 comes out into the intermembrane space in exchange for an incoming acyl carnitine with the help of a facilitated diffusion by the enzyme translocase. So coming to the beta oxidation proper, today we will discuss about the beta oxidation of the even chain saturated fatty acids. Actually, 4 reactions are involved. The first one is the formation of trans delta 2 enoyl CoA. The acyl CoA reacts with the enzyme bound FAD to produce trans delta 2 enoyl CoA and enzyme bound FADH2. The enzyme participating is called the acyl CoA dehydrogenase enzyme. In the step number 2, the trans delta 2 enoyl CoA is hydrated by the enoyl CoA hydratase enzyme to produce the L-beta hydroxy acyl CoA. In the third step, the L-beta hydroxy acyl CoA is converted to beta ketoacyl CoA with the help of the enzyme L-beta hydroxy acyl CoA dehydrogenase, which requires NAD+ as its cofactor. In step number 4, the beta ketoacyl CoA, which is formed, that can be shown from this particular slide also, is acted upon by the enzyme fatty acyl thiolase and it reacts with the coenzyme A to produce acetyl CoA and acyl CoA, which is shortened of 2 carbon atoms with regard to the starting fatty acid. So this particular slide will actually show you how these 4 steps of beta oxidation are taking place. The first step is the oxidation. The second step is the hydration one. The third step is again an oxidation step. And the fourth step is a thiolysis step. So the overall reaction for one turn of beta oxidation of fatty acids is shown in this particular slide. And for instance, if we give the example of palmitoyl CoA, then there will be the formation of myristoyl CoA. Palmitoyl CoA has got 16 carbon atoms, whereas myristoyl CoA has got 14. Now, the fate of the products of beta oxidation of fatty acids is that the fatty acyl-CoA is produced, which is actually the substrate for the next round of beta oxidation, whereas acetyl-CoA, which is generated as the second product, enters the TCA cycle for their further oxidation. Now, coming to the energy or the ATP yield from the beta oxidation of fatty acids, which is very important from our metabolism point of view, is that one round of beta oxidation actually produces one NADH, one FADH2 and one acetyl-CoA molecule. This acetyl-CoA will enter the citric acid cycle, and with the participations from ETC and oxidative phosphorylation, it will release more and more of NADH and FADH2, all of which will be converted to the energy equivalents of ATP at the end. So as can be seen from this particular slide, if we take the example of the beta oxidation of palmitoyl-CoA, then there is a release of 129 ATP molecules. Actually, 131 ATP are produced, but we have to subtract 2 ATP molecules as an energy cost for the fatty acid activation. So the net ATP yield is 131 minus 2, that is 129. And the overall equation for palmitic acid oxidation is shown in this particular slide. Today we have undergone a revision lecture on how fatty acids are activated, how the fatty acids after their activation are transported, and how they are beta oxidized and they enter the tricarboxylic acid cycle, the ETC and the oxidative phosphorylation to give our energy currency, which is ATP. Thank you.